Hello, I'm Terry Dermody. I'm in the departments of pediatrics and microbiology and molecular genetics, as well as the program in microbiology and immunology here at the University of Pittsburgh. My laboratory is located in the Rangos Research Center at the Children's Hospital of Pittsburgh in the Lawrenceville neighborhood. Our laboratory is interested in viral pathogenesis and the development of antiviral vaccines. Most of our work concerns reovirus, a cause of encephalitis in very young infants, and recently been described as a trigger for celiac disease or gluten-sensitive enteropathy. We're focused largely on how reoviruses home in the host and target specific cells and tissues. We find these steps in the virus-host interaction fascinating. How viruses traverse anatomical barriers and evade sentinel host defenses in order to dock onto susceptible host cells, often at a distant site from the initial site of inoculation. We've studied viral receptors and identified several that reoviruses use to bind to particular cells and tissues. We're also interested in the entry mechanisms, mechanisms by which the virus internalizes into cells to launch an infectious cycle. Most remarkably, viruses rewire intracellular circuitry to create whole new organelles within the cell. We call these viral factories, and it's in these factories that viruses concentrate all of the replication machinery, and in so doing, enhance the efficiency of viral replication and also shield certain components of the virus from recognition by intrinsic cellular defenses. These factories are exceedingly efficient, such that in most cells, a single viral particle will give rise to thousands and thousands of viral progeny. Recently, we've discovered a new mechanism whereby reoviruses egress infected cells using a vesicular sorting apparatus derived from modified lysosomes. These modified organelles collect progeny virions, traffic them to the cell surface, and release the progeny particles by a reverse budding mechanism. Most interestingly for us is how reoviruses traverse in neurons and cross synapses to infect subsequent cells within the nervous system, and in so doing damage these cells, leading to the process of encephalitis. One of the newest projects in our laboratory concerns how viruses can trigger celiac disease. And as we've discovered, some reovirus strains, when they infect mice at the same time that a new food antigen is introduced, can serve to block the development of immunological tolerance to the new food antigen. And in fact, the mouse immune system recognizes this new food antigen as foreign and generates an immune response against it. And this is exactly the mechanism that we think is operant in celiac disease. And through our studies of understanding how a viral infection of the intestine, which may not produce any symptoms at all, leads to a failure to develop oral tolerance to food antigen, might give us clues to the cause of celiac disease and might uncover some potential avenues for therapy. Students in my lab gain expertise in genetics and biochemistry and cell biology and work with a terrific team to learn to become scientists, to develop their own projects, and communicate their findings through both oral and written presentations. They enjoy their time in our lab and then move on to a variety of postdoctoral opportunities, some of them in science, some of them in industry, and some of them in other careers. Our team is terrific and truly makes all of the work that we do possible. If you'd like to learn more, email me at Dermody, Dermody, at chp.edu, and we can make some time on the phone or by a Zoom, and I'll look forward to meeting you. Thanks much for your attention.